Welcome everybody to the Biology 1 division. We have a total of 5 presentations today in this division. At the end of each presentation, we'll take questions from the judges and then the audience. You may ask your questions through the chat or use the Zoom raise hand feature and then wait to be called on. Before we begin the presentation, can the judges please briefly introduce themselves and mention their research field? So Debamita, you can go ahead and go first.
Okay, so hi, my name is Dr. Debamita Paul. I am a senior postdoc with Professor Jong-Hoon Min in the Department of Chemistry and Biochemistry. I work on structural studies, mostly structural studies by macromolecular X-ray crystallography, which is a technique to look at structure of large biomolecules, such as protein, DNA, and these days people are also looking at in a bigger context. So we specifically work on DNA repair enzyme. I don't know how much time I have. You can stop me when when you think it's too much. So just a couple of lines. Yeah. These enzymes are these proteins are very important because they are playing vital role in DNA repair. And if DNA lesions are not repaired, that leads to cancer. So by looking at the structure, we want to know how they work. And specifically, we want to use to modulate their function so that, we can have more insights about how to go towards, like different biomarkers, etc., of these cancer pathway. So that's what I do, and I'm very excited to be a part of this conference. So good luck to everybody. Very excited. Yeah.
Thank you. Okay, and Otis, you can go ahead and introduce yourself too.
Good morning, everyone. My name is Otis. I am a fifth-year graduate student at Baylor. I have spent, did my thing, did I mess up?
I think your camera just turned off. Okay, I think you're good.
Okay, something just turned on, that's weird. So recently, the last couple of years, we've been studying, so I think everyone here is probably very familiar um, with phosphorylation, right? So like how phosphate groups are added to like tyrosines and serines and threonines and stuff like that for signaling processes. The exact same thing exists for sulfur and cysteines, right? So instead of phosphorylation, this is per sulfidation. So adding sulfur to cysteines or just changing the oxidation state of cysteine to add 1 or 2 or 3 oxygens, and both changing the protein structure shape, but also as a intracellular signaling pathway. So we've been studying a lot of small sulfur molecules in cells. That's kind of the bioanalytical portion of it. And to kind of understand how like H2S, the small sulfur, HOSH, HOSOH, so 1 or 2 oxygens added to it, or things like HSSH or HSSSH, multi-polysulfides. We see all these being made in cells. And so we're trying to just begin to understand how all these small molecules, which are made endogenously in cells, how that affects persulfidation or cysteine redox statuses for like signaling or maybe later protein structure and stuff like that.
Very cool stuff. Thank you guys for sharing your research with us. Okay, so Dev and Olivia, our first presentation is brought to you by them. So if you guys can share your screen, and when you guys are ready, I will start your timer.
So hopefully you can see our presentation. So hello, thank you all for allowing us to present to you today. My name is Devang Thakur.
My name is Olivia Tolliver.
The topic of our discussion today will regard the inhibition of NF-kappa B in the lungs and how it potentially protects mice from bleomycin-induced lung fibrosis.
To start us off, what is pulmonary fibrosis? It is a debilitating disease that results from injuries to lung parenchyma. The proliferation of fibroblasts and extracellular matrix proteins inhibit normal lung function. the fibroblasts become active and evolve into myofibroblasts, which proliferate in the lungs. These myofibroblasts are linked to the structural alteration caused by remodeling of the ECM, leading to tissue stiffness and impaired lung function. At the moment, treatment regimens for pulmonary fibrosis are very limited, as the two FDA-approved drugs that are currently used to treat pulmonary fibrosis only slow down the fibrotic process instead of stopping it. So identifying key molecules are required for the development of new targeted therapeutics.
Now knowing what pulmonary fibrosis is, the literature also shows that inflammation plays a critical role in pulmonary fibrosis. In the setting of inflammation, NF-kappa B has been shown to be an important modulator for inflammation. NF-kappa B is a pleiotropic transcription factor, which regulates a set of genes responsible for both innate and adaptive immune responses. Apart from its vital role in immune modulation, NF-kappa B contributes a critical role in pathogenesis of several lung diseases, including pulmonary fibrosis. However, the role of NF-kappa B in BLM-induced pulmonary fibrosis is unknown.
So additionally, a small conserved non-coding RNA designated as microRNA was implicated in many vascular pathologies, including pulmonary fibrosis. Therapeutic strategies based on the modulation of microRNA hold a great promise in disease pathologies.
There is still little understanding of the underlying molecular mechanisms of pulmonary fibrosis. A major gap remains in understanding the therapeutic benefits of microRNAs and NF-kappa B linked microRNAs during pulmonary fibrosis. Regulation and expression are key unknown factors. Based on this information, we hypothesize that NF-kappa B inhibition in the lungs mitigates bleomycin-induced pulmonary fibrosis.
To examine our hypothesis, we used the bleomycin mouse model. Age and sex-matched transgenic mice from a C57BL6 background served as our control group, and 8 to 10 week old mice were used in experiments. Our wild type is our control group, 2M is our transgenic group, and those same groups injected with bleomycin created our two other groups, the wild type bleo group and the 2M bleo group. A single bolus of bleomycin was instilled into the lungs tracheally. Wild type and 2M mice were then sacrificed 21 days after the bleomycin treatment, and their lungs were collected. All of these studies were conducted with the approval of the IACUC at Baylor University.
MicroRNA was then isolated from the lungs of wild type and 2M mice. MicroRNA was performed and analyzed by LC Biosciences, and then RNA was extracted from all mice groups. All experiments were performed at least 3 to 6 times to ensure validity, and data was expressed in means and standard errors. To begin our results, here in figure 1, you will see the 5 genes we have chosen: COL1, CTGF, TGF-beta1, ACTA2, and MMP2. These 5 genes were chosen for a study because they have been shown to be involved with pulmonary fibrosis. As you can see in these graphs, the 2M BLM group shows significant reduction in all genes in comparison to the wild type BLM group, indicating that these genes have an interaction with pulmonary fibrosis and ECM buildup when NF-kappa B response is inhibited in our BLM-treated transgenic mice group, 2M BLM. And just in case it's difficult to see, from left to right, the first column is wild type, then wild type BLM, then 2M and 2M BLM.
Furthermore, it has been established that the pro-inflammatory cytokines IL-6, IL-1 beta, and RELA work with fibrotic genes to advance pulmonary fibrosis. The expression of these genes was increased in the wild-type BLM group compared to the untreated wild-type group, and demonstrate a significant downregulation in the 2M BLM group. In addition to fibrotic genes and inflammatory genes, we are interested in looking at BMR1, CAV1, and EDN1, which were reported to have a role in pulmonary fibrosis. However, their role in the bleomycin-induced pulmonary fibrosis model is unknown. These genes were reduced in BLM-treated wild-type group compared to our wild-type group, and were restored in the BLM-treated 2M group. The restoration of these genes suggests that they are regulated by NF-kappa B. Additionally, the expression of apoptotic genes such as BAC1 have shown to be involved in fibrotic processes. Our results indicate that BAC is increased in the BLM wild-type group and reduced in the 2M BLM mice, In examining these genes, we have shown that they all play a part in the mechanisms of pulmonary fibrosis.
Now, in addition to the gene expression profiling in BLM-induced pulmonary fibrosis that we've shown, this next slide will show you the microRNA array analysis, and it displays a heat map which provides a graphical representation of fold regulation expression between two groups overlaid onto the PCR array plate layout. This array shows the dysregulation pattern of microRNA in green and red colors. From this, we selected two microRNAs based on the bioinformatics algorithm tools, like TargetScan and microRNA-base. The reason we chose MIR355P and LET7A5P are because they may be involved in fibrotic gene modulation and regulated by NF-kappa B as well. Here, this slide shows the expression of MIR355P and LET7A5P in a BLM-induced fibrosis model. Our data suggests that both microRNAs are upregulated in wild-type BLM mice and were significantly reduced in 2M BLM mice, indicating a link between NF-kappa B microRNA and pulmonary fibrosis.
In addition, we chose MIR5035P and LET7A5P because, as you can see in this figure, these microRNA target BMPR1 and EDN1, both of which play a significant role in pulmonary fibrosis. When these two microRNAs are upregulated, their target genes are reduced, meaning that these microRNA inhibit the expression of these genes. Our results demonstrated for the first time that the inhibition of NF-kappa B in the lungs repressed BLM-induced lung fibrosis. Several fibrotic hallmark genes, including COL1A1, COL3A1, CTGF, MMP2, and TGF-beta1 were significantly reduced in BLM-induced 2M mice compared to our wild-type mice. The study also showed for the first time a significant reduction in CAV1 and BMPR2 in BLM-induced pulmonary fibrosis, which was restored in 2M mice, indicating an NF-kappa B mediated regulation.
Finally, we have identified several dysregulated microRNAs in BLM-treated wild-type mice, and the current study validated LET7A5P and MIR5035P as potential microRNAs in pulmonary fibrosis. Essentially, these microRNAs were upregulated in wild-type BLM groups and were reduced in 2M BLM groups, suggesting their possible regulation by NF-kappa B. Our study identified the importance of NF-kappa B, MIR5035P, COL1A1, BMPR1A axis in BLM-induced lung fibrosis. The cytokine released followed by end-MT may provide a new mechanistic information for the progression of lung fibrosis. Our data further suggests that LET7A5P and MIR5035P contribute an important role in lung fibrosis and may be considered as a possible therapeutic target.
While the work was conducted within Dr. Gupta's lab in the biology department, we would also like to acknowledge Dr. Dwayne Simmons for his support in providing its laboratory facility to perform experiments and his team members to assist us whenever needed. We would also like to acknowledge Dr. Ryan Stoffel for his aid in vivarium handling, surgery, and installation. This study was partly supported by the American Heart Association.
This will end our presentation, and at this time, we would like to take any questions or comments.
Thank you, Deb and Olivia, for your presentation. Do any of the judges have any questions?
First of all, nice presentation. It was good. Thank you. I have maybe it's a basic question. So what is the difference between lung fibrosis and bleomycin-induced lung fibrosis? Why did you choose the bleomycin-induced ones for your study?
So bleomycin was an easy way to induce fibrosis within the mice. And bleomycin is a known cancerous
Effects of bleomycin-induced lung fibrosis?
It would be our wild type non-treated group and then our wild type treated group.
Okay. Okay, all right, thank you.
Oh, we can't hear you, I'm sorry.
Otis,
Yeah.
Yeah,
I'm
we
not
can't-
sure if I'm not sure if your audio's not working, but we can't hear you.
No.
No.
If you'd like to, you could leave the question in the chat, and then I believe Dev and Olivia could answer it from there. So for those who can't see the chat for some reason, I can just read the question. So Deb and Olivia, you can see it as well. How does NFKB regulate miRNA through transcription or translation control or some other mechanism?
I believe it's transcriptional control because NF-κB is a pleiotropic transcription factor, and it handles the genes for the inflammatory cytokines, the fibrotic genes. It regulates all of those genes, and in response to like plaque buildup and ECM buildup. So by
all of the genes that we discussed, the fibrotic genes, the cytokine, the apoptotic genes, and including the ones that we were unsure exactly how they contributed to pulmonary fibrosis, all of them are regulated by NF-κB through transcription. So our model examined all of those different genes because they connected to NF-κB.
Okay, thank you, Deb and Olivia, for your great presentation. Next, we will have Kathleen, who is also from Baylor University. So Kathleen, I believe you should be able to share your screen. Let me just pause the recording real quick, and then just give me a couple of seconds, and then you can go ahead and get started.
Hello, my name is Keely McKay. I am an undergrad research assistant at Dr. Sim's lab at Baylor University. We are a medical entomology lab that primarily focuses on the study of diapause and the sex determination of Culex pipiens mosquitoes. This project is the analysis of temperature-dependent genitalia rotation in male Culex pipiens mosquitoes. Mosquitoes are by far the most deadly animal in the world, killing more people than any other animal, including things like snakes, dogs, other humans, and other arthropod vectors. A big reason for this is because of how widespread mosquitoes are. They can adapt to almost any climate, so you can find them all over the world. And they also are vectors for very dangerous diseases, such as malaria, West Nile, dengue, chikungunya, all of which pose a great threat to human health. This map shows the distribution of Culex mosquitoes across the world. And if you look in the blue, that is the distribution of Culex pipiens, which is the specific mosquito we are looking at. And as you can see, they seem to be able to adapt to any climate, even going into colder weathers. And that makes them particularly dangerous and why we need to study them. They're also extremely dangerous because the diseases they carry, West Nile, chikungunya, and several types of encephalitis, none of which have any vaccines or cures or any proper treatments. So they pose a really big risk to human health. Another thing that makes the Culex pipiens very dangerous is their ability to crossbreed within the Culex pipiens complex. So if you take a Culex mosquito who doesn't show signs of diapause and you breed them with Culex pipiens, which does show signs of diapause, their offspring will now be able to show signs of diapause, and it'll increase their survivability rate in different climates. This is the mosquito life cycle. Under normal conditions, it'll usually take about a week for the cycle to complete. However, it is temperature dependent. So the colder the weather is, the longer it'll take it to complete, and it can take upwards of a month, depending on how cold it is. In this study, we're specifically looking at the stage in between the pupal stage and the adult stage, right after eclosion. Right after eclosion, in male Culex pipiens, they undergo a clasper rotation, and that is what we were looking at in order to sterilize these mosquitoes. As I previously said, this project is the analysis of temperature-dependent genitalia rotation in male Culex pipiens mosquitoes. The overall objective was to determine the effect of temperature-dependent clasper rotations to Culex pipiens mosquitoes. It's very important to know if the rotation is temperature dependent, because in order to properly sterilize the mosquitoes, it must be done either during or before stage 2 of rotation. So if temperature infects how quickly it rotates, we must know so we can sterilize the mosquitoes in time. Because of how widespread and dangerous mosquitoes are, there's a great need for population control methods like sterile insect technique. However, techniques like this, their efficiency is dependent on the time of sterilization, which is ideally before or during stage 2 of rotation. So for this, we need a greater understanding of the male maturation cycle of Culex pipiens mosquitoes, specifically how their clasper rotation works and the rate at which it occurs. Under normal conditions, that clasper rotation should happen completely within the first 24 hours of eclose. However, as we'll see later on, it is temperature dependent and can take longer. What clasper rotation is it's an evolutionary need to allow genitalia to be in a proper direction for mating. So for male Culex pipiens, they must undergo a 180-degree clasper rotation in order to properly grasp onto a female to reproduce. If they don't undergo this 180-degree rotation, they will be almost impossible to be able to grasp onto the female and reproduce, therefore making them sterile. Typically, this rotation will happen within the first 24 hours of eclose. However, you have to sterilize before stage 2, or else there is a possibility that the males can still grasp on and reproduce. To start off our experiment, we first had to sex the pupa, as we only wanted to look at male pupas since they're the only ones who show the phenotype for claspers. So in order to do this, we took the pupa and we put them under a microscope, and typically the females, which are on the left, are larger than the males are, and they also lack a hook-like structure on the ends of their tails, while the males are typically smaller and have the presence of this hook-like structure. From there, we took the pupa and we put them into cages at various temperatures. We did 25 degrees, 22 degrees, and 18 degrees Celsius. And we let the mosquitoes eclose. And every 4 hours, we would take them out, image and analyze them to see how far along they were in their stages of rotation in order to see what rate the rotation was happening at each temperature. These are the images we took. So image A shows a picture of a mosquito on its back during stage 0, so before the rotation starts. And what we're looking for is the center line that's kind of dark in image A. It's perfectly centered and in line with each other. And we will see that begin to move as the clasper rotates. Image B also shows stage 0, but it shows a side image. And as you can see, that line is now on the left. Image C shows the mosquito on its back during stage 1 of clasper rotation. And as you can see, that line at the very end has now shifted or 90-degree rotation. Looking at is now fully on the side of the mosquito. And image F is the side view of stage 2. And as you can see, it looks extremely similar to the stage 0 of the back in image A. However, this is on its side. Image G is the picture of the mosquito on its back during stage 3 of rotation. And that black line is now becoming visible again on the right side as it turns to line up once more. And image H is the side view of the mosquito during stage 3. And as you can see, the clasper, if you compare it to image D, is now facing the opposite way and is almost completely rotated. Image J shows the final stage, stage 4 of the mosquito on its back. And you can see that dark line has finally lined up again. And image K shows the side view of stage 4. And if you compare it to image B, which was stage 0, you can see the clasper has now made its full 180-degree rotation. For the statistical analysis, what we were looking at is the comparison between the rates and speeds of clasper rotation at different temperatures. So what we were looking at is how significant the proportional differences were. And the way we measured the significance was if the p-value was less than 0.05. And so the trend that seems to have occurred is as time went on, the significance in the proportional difference seems to increase as each hour continues. As I previously showed, this is the experimental setup we used with the mosquitoes being in separate cages at separate temperatures. So for this graph, we are specifically looking at the 25°C cages. When looking at this graph, you can see that the first measurement, which was at hour 2, we have a few mosquitoes who still hadn't begun to rotate and were in stage 0. However, the majority of these mosquitoes had already entered stage 1 rotation. By hour 6, most of them had already moved on to stage 2. A few were left in stage 1 and a few had continued on into stage 3. By hour 10, there was still a significant amount who were in stage 2, but now the majority of the mosquitoes were in stage 3 rotation, and a few had even made it into stage 4 rotation. By hour 14, still a few remained in stage 2. However, the majority remained to be in stage 3, and a few more had continued on into stage 4. By hour 18, the majority of the mosquitoes had now entered stage 4, and a few were left in stage 3. And by hour 22, all of the mosquitoes had undergone a full rotation. This once again shows the experimental setup we used with the mosquitoes being in different cages at different temperatures before we analyze them. For this graph, we were specifically looking at the 22°C mosquitoes. So as you can see, it looks fairly similar to the 25°C ones. However, there is more stage 0 in the beginning at hour 2, but there's still a more significant amount of stage 1 at hour 2. So you can see it's going a tad slower than the 25°C graph was. By hour 6, the majority had gone into stage 1 already, and quite a few had moved on into stage 2. By hour 10, a few mosquitoes also still remained in stage 1, which is a bit slower than we saw before, but the majority had already moved on into stage 2, and a few had moved on into stage 3. By hour 14, we still had a lot of stage 2, but the majority had actually moved on to stage 3 at this point, and quite a few had moved on into stage 4. By hour 18, stage 2 still remained. However, the majority still remained at stage 3, and quite a significant amount have moved on to stage 4 at this point. By hour 22, now the majority of the mosquitoes were at stage 4, but a few were still left in stage 3, and it took 26 hours for the mosquitoes to undergo full rotation. Once again, this is the experimental setup we used, where the mosquitoes were in the cages before we analyzed them at different temperatures. So for this graph, we are looking at the 18°C cages. And right off the bat, you can tell that this graph is significantly different than both the 22 degrees and the 25°C, as it goes significantly slower. So by hour 2, you have the majority of the mosquitoes not even having started their clasper rotation yet and being in stage 0. A few were in stage 1, but not a significant amount. By stage 6, now the majority had turned into by hour 6, now the majority had gone into stage 1, but there were still quite a few left in stage 0. And in hour 10, we still saw some that hadn't begun the rotation in hour 0, but the majority were now in stage 1 and a few had moved on into stage 2. By hour 14, the majority have now moved on into stage 2, but quite a few were left in stage 1. And by hour 18, almost all of them were in stage 2 at this point, but a few had moved on to stage 3 already. By hour 22, it's very similar to hour 18. However, a few more had moved on into stage 3. By hour 26, the majority of the mosquitoes had now entered into stage 3, and a few had entered into stage 4. By hour 30, quite a few mosquitoes had entered stage 3, and most entered stage 4. And we see the same trend going into the 34 hours, and by 38, all had made a complete rotation. So as we can see, the clasper rotation is temperature dependent, and as the temperature decreases, as does rotation speed. So the next step in this line of research would be to study the genes that cause clasper rotation and try to knock them down using RNAi in hopes to completely stop clasper rotation. Questions?
Thank you, Kaylee.
Sorry, this is a little out of my field. Maybe I'm asking a very basic question. Do we know the reason why temperature affects the clasper rotation? Like, we know it is affecting, but what's the, thought? I mean, why should it affect?
We're not positive why, but we're assuming it's because ideally, so the reason we picked the temperatures was the 25 degrees Celsius is the ideal temperatures mosquitoes will live at. So out in like the wild, 22 degrees Celsius was our control at room temperature, and 18 degrees Celsius was a diapause temperature.
Mm-hmm.
So we assume that because they typically live in the 25 degrees Celsius, they've just adapted to have a faster rotation speed at that level than they would at lower temperatures.
Okay. And do we see any similar temperature effect in the female mosquitoes, maybe in some other form, or it's just typically for the male we are seeing this?
It is typically just for the males because the males are the only ones who show the phenotype for claspers.
Oh, okay. I
Yeah.
see. Okay. Thank you.
Okay, so I guess one question I would have is, so 25C is like 77F, seems fairly mild compared to things around the equator, especially like India or Africa. And then a corollary question to that, which is that I would assume that temperature actually fluctuates every day, right? So it's like hot at like 1 or 2 p.m. and then it gets colder. So do people ever have a sort of, temperature modulation, sinusoidal or whatever, when they do things like this, or always just keep it steady?
I think from the research I've seen, we always keep it steady. And the reasoning why we did 25 degrees Celsius and it was more mild is because Culex pipiens are mosquitoes that tend to inhabit more colder temperatures because they undergo this adult diapause, which allows them to almost hibernate during cold temperatures to survive. So typically you'll see these in like, if I go back to the map, you can see they tend to inhabit a lot of places like near Europe. So there is some that go into the hotter temperatures, but for the majority, the Culex pipiens tend to stay near the colder weathers because that's where they can survive without competition from other mosquitoes.
Right. Okay, and then my second, I don't know what number this is. So my last question is, okay, so mosquitoes are dangerous. Being able to sterilize them would help with disease control, but you need to catch them when they're in the pupa stage before they become adults. So how does this translate to actual like how can you find, I guess what I don't understand is that if you want to control mosquito population, how can you go out into the wild and find the pupa? You know what I mean? I guess I just don't understand, like by the time they're already adults in the wild, it's already too late because they've already had the genital, whatever the word was.
So ideally, this research is more of a preliminary kind of test of concept research. So the next steps are going to be to look at RNAi to see if we can knock down the genes that cause clasper rotation. Because with this method, we do have to catch it in the pupal stage before the clasper rotation completes. But if we're able to knock out the genes that control clasper rotation with RNAi, then we can completely stop clasper rotation altogether. And from there, you can even go into CRISPR-Cas9 if we can see that the knockdown works and just completely remove those genes altogether. And then we would take like a sample and release them out into the wild. And ideally, they could kind of outcompete the other ones for resources to help bring down the survivability of wild type.
All right, cool. Yeah, that was my only question or questions.
Okay, thank you, Kaylee. Great presentation. So before we move on to the next presentation, Otis, Debamita, do you guys need some more time to fill out the forms?
Like I need like 1 minute.
Okay. Debamita, are you good as well? Or do you need some more?
Yeah, like 1 minute is good. Thank you.
Okay. So in the meantime, Faith, do you want to get your presentation up? You can share your screen.
Sure, yeah.
Okay, I'm done whenever you are, Debamita.
Yeah, I'm good.
So our next presentation is going to be by Faith Cox, and she is from Tarleton State University. You can go ahead and get started whenever.
Okay. Hi, I'm Faith. I'm presenting my presentation, the development of a triplex quantitative PCR assay for avian retroviruses. Reticular endotheliosis virus, or REV, is an avian retrovirus that infects the B cells of galliforms, so turkeys and chickens, anseriforms, ducks, and passeriforms, which are sparrows and songbirds. Infection is associated with anemia, immunosuppression, neoplasia, so uncontrolled cell growth or cancer, runting, and feathering abnormalities. One particular bird of interest that REV infects is the Atwater's prairie chicken. The Atwater's prairie chicken is one of the most endangered birds in North America, with only about 100 adult birds remaining in captivity at Fossil Rim and the 2 zoos that do conservation efforts. In addition, only about 42 birds remain in the wild, and every time there's a hurricane that hits their breeding area, all of the eggs get lost from the breeding season. For example, after Hurricane Harvey, all eggs from that breeding season were lost. During a 24-month period, at least 31 captive birds were infected with REV. Seeing as there's only 100 adult birds left, you can see how 31 captive birds being infected is problematic. And after infection is detected, nearly all birds do have to be euthanized. In the past, we surveyed Texas to determine if wild turkeys could possibly be acting as a reservoir for REV and possibly transmitting the disease to the Atwater's prairie chicken. In total, we found 5 REV-infected Rio Grande turkeys from 331 sampled wild turkeys from 18 counties. These are denoted by these purple circles filled in with a number of wild turkeys found that were positive for REV. In addition, we were able to screen 88 imported wild turkeys from West Virginia, and we found 1 positive REV-infected eastern turkey that was imported, denoted by this purple box. These purple circles denote the Atwater's prairie chicken sites at the time of our survey, and this purple and orange circle denotes where our lab is. In addition, that is also the location of Fossil Rim. Another avian retrovirus that I'm investigating is lymphoproliferative disease virus, or LPDV. LPDV and REV present with the same clinical symptoms and are actually indistinguishable from each other on presentation. It's been found that eastern wild turkeys have about a 47% prevalence of lymphoproliferative disease virus, and the prevalence varies between states, between 26% to 83% of birds being positive. It has been detected in states neighboring Texas, but not yet in Texas. In addition, we are interested in LPDV because it has been found in West Virginia, and we've already previously found an REV-infected imported turkey. So we believe it feasible that we have also imported LPDV-infected turkeys. To begin, I designed my assay to target the pan-avian GAPDH gene to act as an internal cellular DNA extraction control. In addition, it targets the REV envelope and the LPDV envelope. I designed my primers and probes initially with the IDT PrimerQuest software. This allowed me to set parameters that targeted conserved regions of the genes. In addition, allowed me to set parameters that required a similar annealing temperature between the primers and probes, required the same amplicon length, and it minimized cross-reactivity between the primers and probes. I further analyzed the primer and probes with the Thermo Fisher multiple primer analyzer to ensure that there was minimized homo and heterodimer formation to ensure that they were in fact compatible for multiplexing. To further ensure that they were compatible for multiplexing, I specifically chose fluorophores that use different channels of the thermocycle that I use. So the REV envelope gene uses the FAM-fluorophore. The LPDP envelope gene uses the hex fluorophore, and the turkey gap DH gene uses the sci-5 fluorophore. Using these fluorophores minimizes channel crosstalk and reduces false positives. If you're unfamiliar with qPCR, here's a general rundown how it works. There's an initial denaturation temperature, which separates the strands of DNA before lowering to an annealing temperature where the primers and probes then bind to the DNA. On the probe, there is a fluorophore, and my probes are also designed with double quenchers. These quenchers act to block the emission signal given off by the fluorophore, and this reduces false positives and reduces premature detection. I have double quenchers which further reduces false positives. After your annealing temperature, you increase to an elongation temperature where the polymerase then synthesizes the DNA strand. It hydrolyzes your probe, which causes the fluorophore to break free of the double quenchers, which then allows the fluorescence to be measured after each round of amplification. To actually establish the reaction conditions of my assay, I started with establishing a temperature gradient. I conducted my temperature gradient starting at 52 degrees Celsius, increasing 0.7 degrees Celsius per row up to 64 degrees Celsius. This allowed me to find the optimum annealing temperature. I did this by running my amplifications out on a 2% agarose tris borate EDTA gel and selecting the band that produced the most intense band while minimizing primer dimer formation. I actually found that temperatures between 54 degrees Celsius to 58 degrees Celsius did not change the intensity of the band much at all, so I proceeded with 56 degrees Celsius. After that, I moved on to establishing my optimum primer concentrations. On my y-axis, I have my base pair ladder labeled. On my bottom x-axis, I have my positive and negative control DNAs controls. And on the top of the x-axis, I have my primer concentrations. I performed a dilution series ranging in 500 nanomolar down to 100 nanomolar for my primers, decreasing at 100 nanomolar intervals. What I did for this was I ran the amplifications with these varying primer concentrations, and then I ran them out on a 2% agarose TBE gel, and I selected the primer concentration that produced the most intense bands while also still minimizing nonspecific binding and primer dimer formation. As you can see, as you increase the primer concentration, the bands get more intense, and that I had no primer dimer formation and no nonspecific binding. So I proceeded with the 500 nanomolar amount. After this, I established the optimum probe concentration. So with qPCR, you add the probe in excess. So you want to add enough because it's hydrolyzed, it's not a reusable reagent. You add the probe in excess. As it gets hydrolyzed, it gets used up. So you add it in excess so that you have more than what you need and you don't run out. If you run out prematurely, then you cannot quantify your target DNA. But you reach a concentration when you've added more probe than you need and you're just wasting it. So this was to determine exactly what that amount was. So I conducted a dilution on my primers my probes ranging in 300 nanomolar down to 50 nanomolar, decreasing at 50 nanomolar increments. On my x-axis, I have my cycle quantification values, and on my y-axis, on my x-axis, I have my probe concentration values. The cycle quantification value is the time that the fluorophore is detected in the amplification, and a lower cycle quantification value is better. So for this, I then compared the cycle quantification values with a one-way ANOVA and then determined the statistical significance with a Tukey post hoc test. This allowed me to select the concentration of probe that caused a statistically significant increase in a statistically significant improved cycle quantification value. From this, I found that the 200 nanomolar concentration was the optimum concentration for probe. After that, I ran my amplifications in triplicate as singleplex assays. This allowed me to determine the expected cycle quantification value of an optimized singleplex assay. When you're optimizing a multiplex assay, you first have to optimize them as singleplex reactions before combining them for multiplexing to ensure that they are not cross-reacting. So on my y-axis, I have my relative fluorescent units. On my x-axis, I have my cycles of both my singleplex and multiplex reactions. The cycle quantification value between the singleplex and multiplex reactions remained consistent, which showed that there was no cross-reactivity found between the 3 concurrent reactions. After this, I conducted a standard curve. The purpose of the standard curve is that it shows your efficiency, your correlation coefficient, and the slope of your reaction. In addition, the standard curve is important when you're actually running a survey, because that is when you can quantify your DNA. On my y-axis, I have my cycle quantification values. On the x-axis, I have the starting log quantity. My standard curve began with a dilution of 1 times 10 to the 8 down to 1 times 10 to the 1. This will be used to conduct a survey of wild turkeys in Texas for the presence or absence of REV and or LPDV proviral DNA, which would be indicative of infection. In conclusion, I developed a multiplex qPCR assay that targets the REV envelope, the LPDV envelope, and the pan-avian gene gap DH. And I found that the optimum reaction conditions were a 500 nanomolar primer concentration, a 200 nanomolar probe concentration, and an annealing temperature of 56 degrees. I want to thank my advisor, Dr. Dustin Edwards, and then our collaborator, Dr. Jesse Meek, for helping me make my box and whisker plot, and the Texas Parks and Wildlife Department for providing the turkey samples. I'm going to go to my first slide for you guys. Do you guys have any questions?
So when you measure the probe concentration, when you optimized your probe concentration, can you please explain that side another time?
Yes. Okay.
So what I see here is the, so we are looking at this CQ value optimization, mean we should be going for the lowest CQ, is that true?
The lowest CQ that provides significance. So what I found was that using the 250 nanomolar probe didn't provide a significant decrease in the cycle quantification value compared to the 200. So using the 250 nanomolar probe was in excess, but it was in excess to the point that there was some being wasted, if that makes sense. It's more cost efficient to use the 200 nanomolar probe, and you still have enough in excess that the reaction is working efficiently and that it has enough to hydrolyze all of the probe that it needs to use, but you're not applying so much that you are wasting a large amount.
I see. And how expensive are these probes? Is it?
The probes are about $300 for 100 micromolar.
I see. Okay. Makes sense to save, I guess.
Yes. When you're working with 3 of them, it starts to get very costly.
Good job.
Thank you.
Yeah, I don't have any questions. I don't know enough about qPCR method development to ask questions about it, but yeah, interesting.
All right, thank you. Yeah, it's kind of a shrouded field. It takes a little bit to learn how to do all of the things.
Right.
Thank you for having me.
Thank you, Faith. So I just wanted to ask, is Thai Vu here? Potentially under a different name? No. Okay. Well, thank you to all the presenters. You did a wonderful job. This concludes the Biology 1